And now it's very means important to obtain and isolate the desired gene, the gene from which we want to get the product. Means it is very important to get the useful gene, otherwise your whole effort will be useless. Okay, this is the means first step, isolating of the gene. The second step is what? The joining of the desired gene to a vector. Okay, vector, when gene and vector meet together, uh, the recombinant DNA is formed. Okay, the vector which act as a means with carrier of the gene. Okay, vector we means usually use the plasmid and the phage. Okay, which act as a carrier. It act as a vehicle, the carrier of the gene, used for the carrying of the gene. Okay, inside the host, the vector along with the recombinant DNA multiply. Okay, and in the end, the gene, the copies of the gene, the desired gene is also multiply, and the its cloning, its clone is formed. Okay, now what is mean by the recombinant DNA? What is recombinant DNA? Okay, the recombinant DNA is artificially created DNA molecule. Okay, when the gene and the vector meets, what we get? The recombinant DNA. Okay, the sequence of the gene and the sequence of the vector meet together, which is means not normally present in the nature. When they two meet, what we get? The recombinant DNA. Okay, a recombinant DNA is artificially created DNA molecule. When the gene and vector bring together, their sequence meet. These sequence, which are means not normally found in nature. Okay, and the third step is the introduction or the insertion of the the recombinant DNA into a host. Okay, what is third step? The in third step, the recombinant DNA is inserted or introduced into a host. Okay, inside the host, when the host cell divides, when the host cell divides, the recombinant DNA also means pass through progeny to progeny. Okay, मीन्स ये रिकम्बिनेंट डीएनए पहले एक ही सेल में हमने इंट्रोड्यूस करवाया, उसके बाद जब सेल्स की डिवीजन होगी तो ये पास होगा मीन्स प्रोजेनी टू प्रोजेनी ऑफ दैट सेल. Okay, after large number of division of the cell, a colony form होगी, and that the each cell of that colony will contain the recombinant DNA. Okay, the gene which carried by the recombinant DNA is now said to be clone. Okay, हमने recombinant DNA को इंट्रोड्यूस करवाया इस सेल में. जब सेल्स की डिवीजन होगी, उसके बाद कॉलोनी प्रोड्यूस होगी. वो कॉलोनी, each cell of that colony will contain the recombinant DNA. Means that gene will be cloned. Okay, इस प्रोसेस को हम क्या कहेंगे? Cloning. Okay, in the end, the third step, the final step is what we means monitor. We see that either the recombinant DNA is taken by the cell or not. हमने इसमें introduce करवाया many times the recombinant DNA is not taken by the cell. The third and fourth step we means monitor and means observe or check that the recombinant DNA is taken by the cell or the not. Okay, we will means in next video we will be discussing the gene cloning in very detail. Okay, this was only the introduction of the genetic engineering and the gene cloning. In upcoming video we will be discussing that what are the restriction enzymes, okay, what are their importance, why we use them, okay, and what sorts of the enzymes are used in the gene cloning, okay, and we will be discussing the whole process of the gene cloning in very detail, how we isolate and how we introduce the gene and how we form the recombinant DNA, okay. We will be discussing in very detail, okay. Further, if we want to get this material from me, you can email me. I will send it to you. I have means detailed material about genetic engineering and the gene cloning. So if we haven't subscribed my channel, please to subscribe for more videos. अगर आपने मेरे चैनल को सब्सक्राइब नहीं किया तो प्लीज़ इसको सब्सक्राइब करें और बहुत से वीडियोस आप तक पहुंच पाएंगे उस ज़रिए से। तो तब तक के लिए अल्लाह हाफिज़।